right, if everyone can have their seats, we're going to go ahead and get started. This is quite a popular session. I love it. If you can't find seats, there's some. Raise your hand if you have a seat open next to you. People are coming in. Great. Fantastic. And hopefully they'll shut the door back there. I am hoping. Okay, we have a lot to discuss today, so we're going to get started. We're a couple minutes late, so we're going to, so I don't get in trouble for things running over, we are going to start and get going. So welcome, everyone, and welcome back to Track 3, Patients as Drivers in Drug Development. We hope you enjoyed the networking lunch break, sponsored by Pfizer. I'm Amy Grover, and I will be your host for this afternoon session. As a reminder, this track is being live streamed, so smile. And I would like to welcome our remote attendees and remind everyone to download our mobile app to use our live Q&A. So we'll have some Q&A at the end of the session. Coming up on stage, or already on stage, is Bo Bigelow, Chairman and Co-Founder, Foundation for USP7 Related Diseases, and Rich Taylor, Professor of Chemistry and Biochemistry at the University of Notre Dame, who will be discussing the best practices in contacting researchers as well as how to bring your rare disease to the forefront for research in this session. Rare disease research, getting your disease noticed. Please join me in welcoming Bo and Rich.
Thank you. So there's three parts to what I want to share with you today. How I got here, and the third part is a very important phone call that I got, and the middle part is the one you really want to, I think, listen to about researchers, which is my secret sauce. So, part one, getting here. This is my daughter, Tess. She is 9 years old, and she has a disease that is so rare it does not have a name. It is called USP7, that's what we call it. It's just the gene that is mutated in her. We do need to come up with a better name, but that's what we call it for now, USP7. She's 9 years old, and she is a very happy girl overall. She's nonverbal. She has autism, epilepsy, and intellectual disability. And when she was first born, we knew that something was a little bit different about her. She was kind of checked out. She was not really engaging with us. There was a lot of time where we weren't sure if she could see us. We weren't sure if sounds were registering for her. A good part of the time, she was just kind of staring straight ahead and didn't seem to engage very much with us or the world around her. And she has an older brother. He's about 3 years older than she is. And he kind of hit all his milestones early. And so when she came along, she was falling behind, and eventually it got to a point where it was undeniable, where she was definitely falling behind her peers, and we knew that something was going on. She also had some bone stuff going on. She had hip dysplasia. Here she is in a full body cast. And over time, there were so many things going wrong with so many systems in Tess that we started to think, well, maybe there's something bigger going on here. Maybe there's some kind of genetic syndrome. So we went through a lot of genetic testing where they had ideas about what it could be, but it looked so much like so many other diseases that one by one we ruled out Angelman syndrome and Prader-Willi syndrome and all these other things that it looked like. And eventually we mapped her genome, and we found out that it was the USP7 gene. But even then, the geneticist said, look, we don't know enough about this gene to even tell you whether this is causing the disorder that Tess has. And we don't know anyone else who has this. So good luck is essentially what they said to us. And so we were in this cloud of unknowing, which people have talked about it all day today. It's a really painful place to be where you have no community, you're alone, you're ice fishing, like in Susanna's example from this morning. And it's just a really hard place to be. And we were in that place for months where we were thinking, well, Tess is the only one of her kind in the world. But then, come on. There we go, okay, sorry. So then I read this article in the New Yorker about another family that was in the same position. It was about a family that they believed their son was the only one of his kind in the world. And the dad of the family, this guy, Matt Might, he didn't want to just let it go. He wasn't willing to do that. And so what he did, he knows that all of us rare disease parents and families and caregivers, we all do the same thing. We all get our kids to bed at night and go to the computer and we Google stuff, right? That's what we do. And some of it is trying to solve medical problems, but a lot of it is about finding connection. Like, is there anyone else out there who has this? You know, is there even one more person that has this same thing? And so Matt knew that that's what we do. And so what he decided is to kind of flip that model on its head. Rather than Googling stuff, what he would do is set up a web page all about his son and let it be a beacon, an internet beacon that other people would find when they Google stuff. So he's a computer science guy, and he was really great about sharing his method. He had this blueprint: do this, put these genetic terms in, and we followed it. We created a web page exactly according to his instructions, and the goal was to find just one more patient, just one more. And we were kind of taking a deep breath and saying to ourselves, you know, it might be, my wife and I said, it might be years before we find even one more person, but we got to try, and you know, let's take a deep breath and put this out there. So we put it on social media, Facebook and Twitter, where we were, and then we asked our friends and family to share it in their networks as well. So it spread to other places where we weren't, like Reddit, for example. So we put it out there. This is the summer of 2015, and we're expecting this might take years. And 24 hours later, we get this email from this guy, the guy in this slide. His name's Mike Fountain, and he says, my name is Mike Fountain. I work at a lab at Baylor College of Medicine in Houston, Texas. I work on this gene. I work on USP7. I know of other patients. Let's talk. So my wife and I get on the phone with this guy literally the day after we put our page together. And he tells us that he works in this lab and that they're about to publish the first paper about USP7. So this paper came out in August 2015. And that was the beginning of a relationship with that lab and with Mike Fountain's boss. His name is Dr. Christian Schaff, and he's the expert on USP7. He published that first paper 4 years ago, and he's written another one since, and we've gone to visit him. Here he is meeting Tess, and she's really interested in his tie. And he's great. He's all in. He's been all in on our disease from the day we talked to him. And so that began a relationship with him that continues to this day. Since then, we've started a foundation, and we've raised a bunch of money that we're contributing to research that his lab is going to do. And around that same time that we found Dr. Schaff, I came to Global Genes. I came here in 2015, in the fall that year, and that was the last time I've been here until now. And while I was here, I met someone who's in this picture. This is Daniel DeFabio. So I met him. He was coming to Global Genes that year. His son has Menkes disease. And he was doing some writing, and at the time I was just starting to kind of put it out there about Tess, starting to tell our story publicly and you know, be comfortable about owning it and putting it on social media. And so somebody got us together, like, you guys are writers, you guys meet, talk. And so I kept in touch with Daniel after Global Genes, and I'm going to come back to him later. He's a really big part of my story, but I just wanted you to see him. So that's how I got here. And now I want to share with you my secret sauce. Now, one thing that happened as soon as we got our diagnosis, somebody pulled my wife aside and they said, you know, no one is ever going to care about Tess's disease as much as you do. And that's, unfortunately, that's life. That's just how it is, right? But not too long after that, I met another rare disease dad named Stuart Seidman, who's here. And Stuart said something to me, which I'll never forget. It was the first time we met, and he said, you know, people don't automatically care, but you are going to make them care. And that's your job. That's what you do now. Your job is to make people care. And I was like, yeah, but how? How, Stuart? Like, what do I do? And he's like, you are going to put Tess in front of these people. And sometimes you're going to be really lucky and you're going to be able to do it in person. Anybody who meets Tess is going to fall in love with Tess and want to help you and want to help your family. But sometimes that's not possible. And so how else can you get Tess in front of people? What does that look like and how do you do it? So what I want to share with you is the tools that I use in order to get Tess in front of people when an in-person meeting isn't possible, okay? And the beauty of these tools is that the weapon, the tool that you can use to use these is your computer, okay? This is what I use all the time, is my laptop more than anything else. All these tools are free and they all work. So the first one is called Gene Cards. And some of you may be familiar with it. This was a really good starting point for me, actually, at Global Genes 4 years ago is where I first found out about it. So if you just got your diagnosis and you only know the name of your gene And you just want to kind of find out what's going on out there, what have people already done, so I don't waste my time and recreate somebody else's work. Gene cards is a really good starting point. You can look it up. You can look up any gene. Come on, there we go. So yeah, there's a search box up top. You just type in your gene. And then once you're in there, this is the results for USP7. Once you're in there, this is all the stuff that's out there in the world about USP7. So that includes if anybody else has a mutation. Sometimes those are listed in gene cards. Any publications that are out there, if anybody's done animal models. So this is a really good starting point, gene cards. And these are the publications that are listed in gene cards for USP7. Now, publications are a really key part of getting your disease noticed, of getting researchers involved and trying to get your rare disease patient in front of them. And there's another tool that, if you're not familiar with it, you really need to be, and that's PubMed. So PubMed is essentially a database of the papers that are out there, about medical papers, right? And it's searchable, again, by gene. So here are the results for USP7. And what you do is every few days, you get on PubMed and you look stuff up and you see, is there any new paper about my gene? You just do that. You do that every day or two because there's always going to be stuff being published and you want to stay up to date on it. And then if you're like me, maybe you have the feeling that you're bothering people. You know, people are the researchers who write these papers, that they don't want to be bothered and they're kind of busy doing their lab stuff and they don't want to hear from you. But in fact, it's the opposite. Because everybody who writes a paper that shows up on PubMed, they got into this to help people, right? And here you are getting in touch with them, because that's what you're going to do, and I'll get to that in a minute. Once you have the paper, you're going to get in touch with the people who wrote it, okay? And you're getting in front of them with an opportunity. You're telling them, I have a real live patient, an actual person who could benefit from the work you just did. You wrote this paper, and it could really help Tess. So let's talk. And that's what I do. I bother people all the time. I'm a total nudge to like all the people that write papers that show up on PubMed. And so what you're going to want to do is for any paper that comes out, the most important thing is to look at the authors. So if you see here, they're listed in a certain order. And I didn't know this. You wouldn't know this unless somebody told you. But the order that they're listed in is crucial. There's kind of this hierarchy, right? So the big kahunas of any paper are always going to be the first author and the last author. Those are the people who, like, it's their lab where the work got done, or they're the ones who got the grant and funded the whole thing. Whatever it is, those two people, first and last, those are the ones that you want to get in touch with, okay? And sometimes you're lucky, like if you look here, so the last author, number 6 there, is listed at the bottom, and there's an email address for them. So boom, right there, if you click on that person, you can get their email address, and then you email them, and you talk to them about your disease, and you get them involved. And sometimes you're not lucky enough to have the email on there. So the first author here doesn't have one listed, but listed there is where they work. So now you have their name, and you know where they work, and you're going to cyber stalk them until you get their email address, and then you're going to email them, same thing. So you're going to get in the habit of doing that, of contacting those researchers. If possible, before you get in touch with them, read their paper. It's a great thing to do. Now, I'm a French major, so I am useless when it comes to science. Anytime I pull up any of these papers, I have like 20 Wikipedia tabs open at the same time, like going back and forth. And that's just what you do. And it's fine. Nobody's looking. Like, you are going to teach yourself so much stuff that you wouldn't even believe it. And every so often, you'll find a paper that's behind a firewall. It's on a website where you got to pay. And so what you can do is email those guys who wrote the paper, email the authors and say, can you please send me your paper? I have yet to contact somebody who's told me no. They're always so juiced. Wow, somebody read my paper, and it's a patient. And yeah, you bet I'd send you a copy. And they do. They email it to you. So you're contacting those authors, you're reading those papers, and you're trying to get them involved. And what you're really doing is trying to kind of widen your circle. Like, it's not enough for them to just do the phone call with you, okay? The idea is to build a tribe, okay? And you're bringing these researchers into your tribe, if they're willing. And that's your goal. You want to make them part of your team. Like Dr. Schaff, who was in that picture. I mean, I didn't really have to do it. He already had the will to do it, and he was interested in working on our gene anyway. But every time I contact a researcher, that's what I'm trying to do. Like, we are doing this thing. We are working on this gene. You could be part of it if you want to come join us. And that's really what you're going to be doing with PubMed. Another huge tool, if you're not on LinkedIn, by the end of this weekend, you should definitely get on LinkedIn. I don't know what it is, but in the last year or so, LinkedIn has like blown up with rare disease stuff going on. I don't know why, but it just seems to be the network of choice for everybody who's working in rare. I can't tell you how many phone calls and meetings and awesome things happen because of LinkedIn and connections that I make on there. And if you're already on LinkedIn, there's this powerful weapon that it lets you do that I really want to point out to you in case you don't know about it. So this is my profile on LinkedIn. And if you look under my name, there's like that little paragraph there. Do you see that rare disease trailblazer it starts with? So that's this thing that LinkedIn lets you do that's called a headline. And the beauty of the headline is that anybody who comes across your profile, and they don't even have to click on your name and look at your full profile, it'll just be under your name, like in search results or anytime you post anything on LinkedIn, your headline is there. And so what you want to do, like my position with Tess is, there's 3 kinds of researchers out there in the world that we need, that are not part of our tribe yet, that we really need on our team. And it's people who are working on autism, and anybody who is working on this thing called the WASH complex. It's like a really sciencey thing that I don't fully understand myself. That's why I need an expert. And then the ubiquitin proteasome system. So I know I need those researchers, so I put it in my headline. They're right there, all 3 of those things, autism, WASH complex, and ubiquitin proteasome. That way, anybody who comes across my LinkedIn profile is gonna see my headline. So it's a really great thing to just build in there and make it clear what your priorities are. And that way, if you're asking for a meeting, you're asking for a phone call or whatever with somebody, they're gonna be like, what's this person really about, you know? And this is a great way to show them. One final thing about LinkedIn, it's also great when every so often you'll come across a paper where it isn't the first and last author, but someone in the middle who was like, this scenario that I want to talk about here, this is somebody who was a student, and she was just in this lab for like a semester, and she was the brains behind the paper that I was so interested in. So when I called the first and last authors, they were like, yeah, I can't really help you with this. I don't really know how it went. She's the one you need to talk to. So, you know, and that was like 3 jobs ago, and I didn't have an email, and I couldn't track her down, but she was on LinkedIn. And so I wrote to her and said, I read your paper, and let's talk, and so she got on the phone with me. So LinkedIn is just another backup in case email doesn't work. Come on. One final thing about secret sauce is on Facebook, it's very daunting to think of doing all this work yourself, especially if there are a lot of publications about your gene, which there are for mine. There's like a new one every couple of days. And it's a really good idea to try to find other families, other people, stakeholders who are in your tribe who maybe they're also hoping to get to a cure or a treatment and they're really motivated. And so what you want to do on Facebook is create a closed group. And that's going to be your kind of HQ, right? That's where you're going to rally people and say, okay, let's split this up. I'm going to do this, you do that. It's just a great way to do it. That way, if you're not familiar with it, it's a great system where if people want to join, you can make it so they have to answer a certain question, like are you somebody who has this mutation, or are you working with a rare disease patient? That way, anybody who's like from outside, who's like a reporter or something, they won't get in, and you can limit it to just families. So that's something that we use for USP7, and it's really effective. Okay, part 3. This is the phone call. So 2 years ago, I get this call from Daniel. Remember him? He calls me up. This is a better picture of him without the beard. So he calls me and he says, you know, I made a film about my son, about Menkes disease, and I've run out of places to show it, so let's start a rare disease film festival. And I just say, no way can I do that right now. It's a great idea, but I'm in the middle of trying to start our nonprofit and trying to get that off the ground. And the big priority of our nonprofit was to find more patients. So Tess was the 8th known patient in the world. And Dr. Schaff, who I showed you a picture of before, he said, find me more patients. Until we get more people, this isn't really gonna go anywhere. So I told Daniel, no, I gotta find more patients. And he said, well, you know, if you're serious about finding more patients, you'll make a film. You should make a film about USP7, and that will do it. That will help you find more patients. So we did. I told him I'm not really a filmmaker, but he said, I am, and I'll help you. I'll do the editing and I'll do the audio. So we made a film together. It's called Tess is Not Alone, a USP7 story. And he convinced me to do this festival. And so 2 years ago in Boston, we started Disorder, the Rare Disease Film Festival. And initially, we had this ultra-selfish goal of showing just the films about our kids, but over time, we developed another goal. And the point of the festival became not just to show our films, but try to curate the audience in the sense that we wanted to get all the right people in the room, like all the rare disease stakeholders who could lead. I'm talking about patients and advocates, researchers, biotech and pharma, get all those people in the room and watching these films, because it's only when all those people are together that you're going to spur collaborations that can lead to cures and treatments. So that's been the goal of the festival basically since the beginning. And it kind of blew up from that point. Like we thought, all right, we'd show our 2 films and maybe one more. Is there even a market for this? We don't really know. Could we get a sponsor perhaps? Like, we just didn't know. It's not exactly like a Will Ferrell film festival where you're going to go and have like the time of your life. This is some pretty heavy stuff, right? Some of these films are about death and dying and pain and really difficult situations that people are facing. So we didn't really know if it would go anywhere, and it did. It blew up. It became this thing that was like, we had over 30 films. It was 2 full days of sold out slates. We had 20 speakers. We had a bunch of sponsors, and we discovered that, yeah, there is an interest in this, and there are a ton of people making films about it. And I think the thing that I keep coming back to about that first year is that I didn't realize until I was there, until we were at the festival, what film really is and what film can really do. Because film is one of the only things we have left in our culture right now that makes us slow down and really listen to somebody's story. You can't, if you're I'm talking about film, not like watching it on your laptop or watching YouTube on your phone. I'm talking about like you're in a theater and the lights go down and there's a big screen, and that's what I mean when I say film. Because in that audience, you cannot check email, you can't multitask, you can't do any of that stuff. You can't pause it, you can't fast forward it and skip to the next part. You gotta just sit there. And so the weight of these stories just is much more impactful. I mean, in the audience, you are just blindsided, and I'm no exception. Like these are films, keep in mind, I have seen these films dozens of times. I picked them. Daniel and I pick all the films. So I've seen all of them. And nevertheless, there I am in Boston. I'm in the back row of the theater. I'm like blubbering into my sleeve because I am as affected as everybody else in the audience by films I've seen before. It's just, there's something about what film does. And so in terms of telling your story and getting noticed, if your rare disease does not have a film, you should really consider making one. And I'm talking about partnering with somebody who maybe has some experience in video production, finding somebody, some friend of a friend, whatever you need to do to network and get your film made. Because Daniel and I feel really strongly that there are 7,000 rare disease, and every one of them should have a film. We can't make all of them, and we can't help you make them all, but we want there to be that many films. There just should. Every disease should have a film. And I think that's because when Daniel and I have sort of a cynical view that we say this all the time whenever we're working on festival stuff, is that awareness is not enough. There's a lot of talk about creating awareness, and what his experience has been with Menkes and what I find with USP7 is there are so many times when somebody learns about USP7 and now they're aware, and then they sail off into the sunset and I never hear from them again, and they do nothing to help me get towards a cure or a treatment. And so I don't think it's enough for there to be just awareness, right? We need awareness that leads to action. And so that's what this festival is about. Yes, we want to make people aware. Yes, we want to tell these stories. Yes, you're going to come to a festival and see a bunch of films about rare diseases that you may never have heard of, but you aren't going to leave it at that. You are going to do something with that awareness. And that's what I'm talking about when I say spur these collaborations and these cures. So we are doing the festival again this year in San Francisco. It's in a couple months. It's on November 9 and 10. There are some postcards out there, and there are some more in the back. in case you need them. And so aside from the festival, we also do a lot of screenings throughout the year. We just did one at Takeda in Boston last week, and we are always looking for more films, not just for our festival, but to be part of these screenings, because the more films get made, the more we all win, because the more people find out about our rare diseases. And so if you have a film or you're thinking about making a film, I really want you to get in touch with me, and we should talk, because I'd really like to work with anybody who wants to make a film. So yeah, the film festival is in San Francisco and in a couple weeks, and in a couple months, sorry. And I hope you can join me there. So thank you.
Thank you very much, Bo. That was inspirational. And myself, I'm a professor of chemistry and biochemistry. I've been at the University of Notre Dame this is my 25th year. So obviously, I come from this discussion from a very different perspective. But I hope what I can give you today is an idea of what happens when people approach me, and what I do, and what I can do, and what hopefully I could be considered a generic researcher, and what perspectives they want. And so I'll have some advice on making that interaction. I'm trying to get the first slide to work. Yeah. So, well, it's not happening. What? This one, right?
Yep, that worked for me.
There we go. It's just a title. I'm bored by that. Well, so I started, as I said, I started 25 years ago. And at the time, I was particularly interested in drug discovery, and so were a couple of my colleagues. And we were talking one day and said, you know, being at the University of Notre Dame, where biomedical research has a significant overlap with our Catholic mission, a focus in rare diseases may be a real opportunity for us. You have to put yourself back to that time of the mid-90s. The pharmaceuticals industry's interest in rare diseases was much less, right? They were still interested in blockbuster drugs, very large market shares. They were not looking for smaller markets. That's changed today. And I'm sure every one of you know that last year, the majority of drugs that were approved, not only was it a large number total of FDA-approved drugs, a majority of them did target rare diseases. Of course, there are 7,000 rare diseases, and with genomics, we're only going to increase that number. And therefore, there's lots of research to do. And of course, my hope is in the research I do is always to find connections between the rare diseases I work on. So any solution could be a potential solution to multiple of these to try to knock down that number. But that effort to think about rare diseases actually ended up culminated in the creation of two complementary centers. The first is the Warren Family Research Center for Drug Discovery and Development, in which we have 28 researchers working in lots of different areas, including rare diseases. But the focus of this center is to help researchers translate their basic research discoveries through the discovery process into the development process, and ultimately into clinical trials, which is always the key goal. So it supports the researchers at the University of Notre Dame to do that. In a complementary perspective, we created a much more focused center called the Center of Rare and Neglected Diseases, the Parseghian Center for Rare and Neglected, and 21 faculty members, some of which have overlap with the Warren Center, work in both basic and translational research associated with a number of different rare diseases. You'll see them listed here, and I'll give you a few examples in somewhat more scientific slides coming up. But it's been really impressive to see what they've done. And all of these rare diseases have come to us from either the Notre Dame community or from the South Bend community. And so it's exactly as Bo was describing, is sort of people reaching out to people who are interested. And what I hope you get from this is we have a number of researchers that are willing to accept things that they do not work on and try to make an impact. Our Bowler Parseghian Center for Rare and Neglected Diseases also has an outreach effort run by Barb Calhoun, who's in the audience and is here this week, who does a number of different things from the development of publishing case reports on various rare diseases, NORD summaries, natural histories, exhaustive literature search, and public awareness campaigns, all with the help of our outstanding undergraduate students. So not only do the graduate students at Notre Dame work on the research associated with these, we often have undergraduates participating in the more public patient advocacy aspects of this. And Barb will, along with two students, Brian Hall and Brian Min, will have a poster on their work about the Bigelow Foundation and preventing childhood blindness at the summit this week. Of course, our interest in rare diseases did have an original why, and those is the picture of that why right here is the three children of our famed football coach, Ara Parseghian. This is Michael, Marcia, and Krista Parseghian, all who passed from Niemann-Pick C disorder, or Niemann-Pick C disease, which is a lysosomal storage disorder. The Ara Parseghian Medical Research Funds got this started, and we have ultimately taken over this, but Notre Dame has had more than a dozen faculty over the past 20 years work on this disease. We organize the annual Parseghian Scientific Conference on Niemann-Pick C research, and just to show you how that's grown, because I think this is an indication on all rare diseases. In 1995, it was a single round table of a couple of researchers, along with obviously interested parents. And in 2019, this conference had 160 attendees, most of which were scientists, but also included patients and patient families. But still, a remarkable growth over that 20 years. During that time, the foundation has raised more than $60 million, and in 2019 supported 15 grants to international researchers in the range of $100,000 to $200,000. We run a number of different events during the year to raise that money, and the organizer, the director of this program, which now resides at the University of Notre Dame, is here, Sean Casson. Here's the website if you wanted to learn more about his efforts there. So this disease is associated with mutation to a gene. Most of it is Niemann-Pick C1, some of it is Niemann-Pick C2. That gene is associated with a protein, and that those proteins are associated with the metabolism of cholesterol. And cholesterol, when there's a mutation to that gene, there's a problem with the protein, and the protein does not extrude cholesterol out into into cells. So it gets stuck in the lysosome. And you can observe this by using what we call the Philippin stain phenotype, that basically shows this glow in these cells that basically say there's too much cholesterol stuck in the lysosome. But when it's treated with a potential drug that can work, it basically allows the cholesterol to get out of the lysosome. So there's actually a couple of compounds in clinical trials right now, one of which is a cyclodextrin derivative, which was showed a long time ago to lead to this phenotypic change. And researchers at the University of Notre Dame have used this assay to discover a number of different compounds that could potentially be drugs based on this. So one of those was done by my colleagues who originally was talking to myself about developing these centers for rare and neglected diseases, and they discovered that histone deacetylase inhibitors increase the expression of genes associated with Niemann-Pick C1 and 2, and then also increase the amount of protein of Niemann-Pick C1, as shown here in a Western blot that says there's more protein when you treat with this compound, and that leads to that same repair mechanism in which allows cholesterol to get out of the lysosome. Notre Dame not only helped support this in a research phase, they also, since one of these HDAC inhibitors was an FDA-approved drug, they helped raise money for a clinical trial. My own lab was inspired by this work, and I work on something slightly different. I work on compounds that are produced by bacteria. So while I'm a chemist and my lab sometimes smells really bad because of those chemicals, my lab often smells like a country garden after a spring rain, because that's what bacteria smells like. So when you smell dirt, you smell bacteria. So one particular bacteria produces a compound called GAX1A, and I was studying this molecule. My lab was looking at this, and because of some of the biological effects of this compound, we thought it might be beneficial for Niemann-Pick C. And so we've done RNA analysis to show actually it affects the concentration of RNA associated with Niemann-Pick C1. And that also leads to the formation of more protein associated for Niemann-Pick C1. So exactly the same phenotypic experiment works, in which you see an overabundance of cholesterol in the lysosome, then you treat with our compound. And at a much lower concentration than the histone deacetylase inhibitors, it leads to an improvement of this phenotype. Now, I wish I could tell you that this compound is now working in animal models associated with Niemann-Pick C. We haven't gotten that far, but we have, based on its mode of action, hypothesized that it could be beneficial for diseases associated with AML, certain forms of AML. And these are basically experiments that show actually this molecule treatment of mice with this compound actually cures their AML. So it is my hope that not only we could push this compound forward for AML, but it also puts us in a nice position to do something in the Niemann-Pick C area. The rationale for showing you these very scientific stories here was the fact that we were willing to work in areas that we had no previous experience. And there's a number of researchers at Notre Dame that are just like that. And there are a number of researchers all over the world that are willing to do this. It's finding them, and how do you find them? Because of our reputation, we were contacted by Matt Wilsey and the Grace Science Foundation, who was working to build a collaborative team and has done so over the past several years. But this was several years ago, a collaborative team to work on NGLY1 deficiency, a disease that his daughter Grace has, along with only 46 known patients. And we basically had a collaboration with Retrophin for 2 years to work on an assay, development of assay for a particular protein called NGACE. And while that collaboration ended after about 2 years, most recently I've started up a new collaboration with an AI drug company, Atomwise, and we've recently identified new leads for inhibiting NGACE and hope to get it into animal models at some point in the future. Sorry about this. Okay, so another story, last two quick ones. I just have 2 slides and then we can get to questions. So I was introduced to a Notre Dame family whose children were suffering from a disease called Cori disease. Cori is a glycogen storage disorder. All I knew about glycogen storage disorders was there were multiple ones, and there's actually more than 13 of them, various classes. But I never heard of Cori disease. But I was asked to look into this, and therefore, what did I do? I basically started by doing, similar to what Bo said, a gene search on this to learn more about the genes associated with this. I used the NIH Genetics Home Reference, but gene cards clearly is something that's very valuable. In fact, probably has a few additional things on it that the Genetics Home Reference doesn't have. And then I moved to Google Scholar and other type of search engines for literature, and I found an interesting correlation that this disease had something to do with glycogen metabolism, and that another disease called Lafora deals with glycogen metabolism in the brain. And there happened to be researchers at the Indiana University School of Medicine who worked on Lafora. So I looked into their work, and I said how their work might actually apply. What they're working on might actually apply to Cori disease, right? So what they basically had done was they basically developed a mouse model for the disease Lafora, which creates bad glycogen, causes the symptoms of the disease in mice. And then they crossbred this mutated mouse with a mutation to PTG, which basically is known to reduce glycogen levels. And the double mutant that had both the Lafora mutations and the PTG mutations basically had normal glycogen and fewer symptoms. So I went to them and basically said, it's known that there is a mouse that's related to Cori disease. If I support you, would you be willing to start moving your project or starting up an additional project through collaborations with me to study whether or not this would work for Cori disease? So they first created the mouse related to Cori disease and basically showed that it had glycogen buildup in the liver and in muscle. And then they crossbred it with the same double mutation to the PGP, creating the double mutant. And although we have only a handful of mice to look at so far, it does have reduced glycogen stores, it has increased body weight, and a decrease in liver size. So it's very likely that this solution, any solution that we find for Lafora, could potentially be beneficial for Cori disease. The problem here is we want to now screen drugs that will be able to do this exact same thing, basically decrease the amount of glycogen. So we've created collaborations with model organism drug companies, initially with Perlara, Ethan Perlstein, and now with James Doyle Modelis, who'll be talking about some of this in tomorrow's session, in which they were interested in making small organisms such as worms and zebrafish. And basically now we're creating them, mutations of them, looking for phenotypes, looking for basically characteristics of the disease, in which now we can do much larger screens of compounds to try to find lead compounds for this. So again, we had never seen this disease before, never knew anything about it, but basically just by doing that literature search and being willing to apply our time to this disease, we've made some nice progress, and we're moving into basically the screening part of this. And just to tell you that this has also inspired someone else at Notre Dame, another Notre Dame family dealing with Shets Disease, an autoinflammatory autoimmune disorder, has now come to us, basically asked whether we'd be willing to spend time on this, and we've created a very nice collaboration to discover something here. It's just getting started, but again, another example of the type of people that are at Notre Dame and the type of people that you can find if you look carefully. So I have some suggestions, just like Bo had. Basically, use the genetic home reference or gene cards to basically learn about the gene and whether or not it encodes for a protein which can tell you more about the biological pathways that are associated with this, and then hopefully even associated with a common disease. All of these are potential ways for you to find researchers, either through people who work on the gene, as Bo discussed, or someone who worked on the associated common disease. The next is to use Google or other search engines, PubMed, to find pertinent literature. Right? And if you have any trouble getting access to that literature, because often literature is behind a paywall, contact me, my email address right here, and we will call it a collaboration, and I will send you these papers. And I really think that Global Genes could do a great service of becoming a network of researchers willing to give out this literature. We'll just call it collaborations, and publishers can't do anything about it. Okay? Thanks. So when you look for researchers, I suggest looking for a record of collaboration. There's no doubt you will meet some people that are not interested. That's okay. The one thing I've learned at Notre Dame, being in academia, is everyone feels overwhelmed at all times, even when people have very small programs versus very large programs. Just some people with very large programs are some of the people most likely to be excited by a new project. Right? Identify researchers with interest in the development of applications and tools. That's the other thing, is platform technologies. So I'm a synthetic chemist. I'm interested in applying my synthesis, my synthetic capabilities, whether it's being produced by bacteria or being produced by the students in my lab. I'm interested in applying chemical technologies to the synthesis of compounds and then using those compounds to learn more about biology and cure disease. People who develop tools like I talked about with worms and fly models of disease are more likely to apply them to new areas. Thank you very much.
It's very interesting.
Thank you so much, Bo and Rich. So we're going to take some questions from the audience as well as I have some questions online. So for those of you watching online, if you want to go ahead and input your question, it does come right to my phone. But if you do have a question from the audience, if you can come to the center here and stand in front of the mic, we can start a line if we do have some questions. But we have some online, and we'll get started. I have one for each of you, actually, and one of them was my question, too. Let's start with Bo. Can you talk about how you came to be comfortable putting your story about Tess out there to the world? Was it something you and your wife were on the same page about from the beginning?
That's a great question. We were on the same page from the beginning, but that's not to say it wasn't difficult and isn't difficult. It's a process, I think, of putting yourself out there, and we've been doing it for 4 years, but I think there's still parts of it that make us a little uncomfortable. You know, the example I always use is I'm in the grocery store, and there's someone there who's like, comes up to me in the produce section and is like, I'm so excited about Tess at school. That was great. Like, I loved hearing about that because I have a podcast that I do every week, so I always tell stories about Tess. And that person says that to me, and I say, I don't think I've met you before. Like, who are you? So it's somebody who knows a ton about us and really personal stuff, and we don't know them at all. And it's unsettling sometimes, but it's just, I think it's just what you need to do. It's just part of owning your disease, and it's just, if you want to make anything happen, my wife and I have always felt like this is just what we have to do.
Okay, let's take a question from our first audience member.
Is
There you
that
go.
There you
good?
go.
More? Okay. I'm a patient, and this is actually my first time at this conference, so it's pretty tremendously special, and thank you guys for being here. It really means quite a lot. I have a rare disorder that's called the MYH9 disorder. It's a mutation on chromosome 22 that predominantly affects non-muscle myosin, particularly non-muscle myosin 2A. Traditionally, this disorder has only been thought about as a disorder of the platelets, that the megakaryocytes aren't forming platelets correctly. But from my own experience and from other patients, as well as from more current literature that's coming out, I'm really coming to believe that this disorder is more of a disorder of the cytoskeleton, and that its basis is more in the misfunction of the myosin 2A and F actin. And that would describe why you have not only giant platelets, but also the other classical symptoms of focal segmental glomerulosclerosis, as well as hearing loss, cataracts, and thrombocytopenia purpura. I, in addition to that, also have a history of 7 different autoimmune disorders, a history of seizures and strokes, and other patients like myself, there's only 300 in the world, have similar symptoms, but none of it is reported in the literature. If this disorder does involve myosin 2A and is a cytoskeleton disorder, it would also account for the autoimmune conditions that you see because of the dysregulation of granules from platelets. But these are all just the things that I'm seeing on my own from looking at the literature and talking with other patients. What I've found is that the researchers, the doctors, and the patients aren't interacting. And none of the patient's experience is driving the research. So if you look up MYH9 online, you won't find anything about seizures, strokes, or autoimmune disorders. You'll only face things about platelets, hearing loss, and kidney disease. So my question is, what do I do to get this research, I guess, started in the right direction?
Well, I'm sorry to hear what you're dealing with. It's amazing what you have already done. So you're already putting together some pieces that are needed for researchers. As a basic researcher, the first thing that sort of hit me from what you were saying that could be a good starting point would be the cytoskeleton. So basic researchers in the cytoskeleton, particularly people who might be able to do live cell imaging. If you use that term, live cell imaging of cells, because the cytoskeleton is a very dynamic process thing, and if that's altered, they would be able to see it. I think that potentially researchers in that area could start the process down a different path, because certainly the two sides look at things very differently, right? One person is the doctors are looking at symptoms and what the patients are dealing with. You've brought it all the way back to the fundamental cellular biology. That's sort of the direction that I come from, and if that will correct how the doctors are viewing this, I think you need more basic research to understand it.
Well, that's very exciting for me to hear, because it actually seems like non-muscle myosin is gaining a lot more importance and interest. And recently, I think just a few months ago, a paper came out showing that non-muscle myosin 2A is intimately involved in red blood cell formation and deformation. And there's also a lot of other non-muscle myosins that are now being found that are 14, 15, and not directly related to my disorder, but are implicated in other disorders. So that basic research seems to almost be there already. It's just not tied into the disorder I have.
Correct. Yeah.
Thank you very much.
Good
I appreciate
luck.
it.
Thank you very much. Let's take one online question real quick. So you talked about the research that you're doing. Can you talk about, from a patient perspective, what do they need in their briefcase when they come to you? What should they have available when they decide to approach you? So that's the first question. The second question is, do you think researchers are getting overwhelmed by, patients as drivers and coming to them with that emotional, appeal?
Who cares? Who cares? Awesome. What to do. Yeah, like I said, some researchers will feel overwhelmed and not interested, but let's find the people who are interested. It's the only way that we're going to solve these problems. There are opportunities. There's no doubt that there's a professional motivation for someone like myself to work in any disease area, right? I pick it because I can support my research. I can think intellectually about things. I can spend my day. I teach, I teach a little bit on the side at Notre Dame. There's a lot of things we do, and it's my job. But at the same time, the work I do in rare diseases, and the reason why I'm here this year and in several previous years is because the patients are the motivators, the families are the motivators, and I think it's very important to remember they are the experts on the disease. I think we just heard that, right? They are the experts on the disease. It's just finding the right researchers that will take what they have to offer and move forward. And I think any bit of information is a good starting point. So some of the people that I've worked with only came with really hope. They didn't come with a lot of science. That's good to know. So it all depends on just who you're talking to. Clearly, the more there is a basis for an understanding of the disease, it's easier for someone to say, I can do something here or not do something. But that shouldn't stop you if you're, Bo's a good example of someone who thought he was alone in the world, and he wasn't. But there are lots of diseases that only have a few people, and that doesn't mean we shouldn't try.
So in other words, don't be overwhelmed. Just find that right.
Absolutely.
It's good advice. Next question.
Hi, my name's Beth, and I'm with the International Wagner Syndrome Association, and I have a question for Bo about your movie. I was wondering if you wouldn't mind telling us what your budget was to do it, and also how you came about funding it. Was it sponsorships? Did you fundraise? How did that work?
When I talked to Daniel, he offered to do a lot of the work for the film for free. And so he did things like editing and a lot of the sort of graphic stuff, like visual pieces of the film he did. I want to say our total budget for the whole thing was maybe $2,000.
That's a steal.
Right. It's a screaming deal. Not everybody's going to have Daniel at their disposal. But I will say this, that almost all of the films that we get, we are looking for films that are like 3 to 5 minutes. So it really doesn't have to be this, blockbuster budget kind of thing. 3 to 5 minutes, and a lot of it, you can really do for cheap. Like with what phones have now, camera-wise. If you know a little bit about lighting and you work with somebody, you can film a lot of it with your phone, and it can end up looking pretty good. So,
where can we see your movie? On your website?
It's on YouTube. If you just look it up, there's a 3-minute version in case you don't have a lot of time, and then there's an 8-minute version. Just look for it. It's called Tess Is Not Alone, a USP7 story.
Thank you.
Thank you.
And next question from the audience.
Hi, my name is Carly Monnier, and I'm with the NR2F1 Foundation. And I just wanted to plug a little bit the film festival. Bo shares the same researcher as we do for the NR2F1, and he's been a tremendous resource. So I hope you've all listened carefully to what he says, because he's a trailblazer indeed, and I've talked to him several times about our foundation and how to get us going. So thank you, Bo, for that. So I'm plugging you on that. And I also wanted to say, just in response to the previous question about the cost of making the film, one of our families has a film in the festival about their daughter with the rare NR2F1 mutation. And she reached out to a student at a university who needed to work on a project. And so everything was done pro bono. And it was very personal. He came to their home. So that's also, students are a great resource. And what else? What was the next thing? That was it, just to plug the film festival, and thank you both for your time.
Excellent. And one last question here.
Hi, my name is Paula. I'm from the Yellow Brick Road Project. We actually showed a film at your 2017 film festival.
Oh, okay.
Myself and my family were in the bottom right picture up there, and your presentation was a
Oh,
little
great.
surreal to see myself up there.
All right.
At the time, my daughter was one of 11 in the whole world. 2 years later, we're at 78. We have St. Jude Children's Research Hospital is doing some of the research that you were talking about, Dr. Taylor, with the mouse models. They have brain organoids, et cetera. But we've spent 2 years trying to go around and get pharmaceutical companies and other researchers interested in moving to the next phase, which would be obviously research into treatment or a cure. And because of our numbers, we have had no luck. Do you have any recommendations other than waiting till the numbers are there? Because as I'm sure you can understand, time is not always an option, and the more time passes, the more likely you are to pass a threshold of whether or not the disorder can be reversed or symptoms can be alleviated. So do you have any recommendation to get that interest if you have such low numbers?
You're talking about for clinical recruitment?
Yes.
Yeah, so I'll first caveat my answer with the fact that I'm not a medical doctor, and therefore clinical recruitment is far from my expertise. But I certainly know it's an issue in rare diseases. So I would say that one possibility is to see about relationships to other diseases that may allow you to add to your numbers or change the clinical trial in some way that would allow you to move a therapy forward that could potentially give you data to help yourself out. That seems to me to be a path that's possible. It's the question of whether or not your disease has the obvious connections to others. But, if a compound basically goes through clinical trials and is shown to be both efficacious and safe, then prescribing it for other diseases may be very easy. So, recruitment is difficult in rare diseases, so whatever we can do to get these compounds tested and approved is number one.
Thank you. Okay, we're right up against the hour, so we will take one more question, but then we'll have to shut down after this.
My name is Jennifer Farabee. I'm here with Carly, representing the NR2F1 Foundation. And you did mention the first question, I believe it was, about putting your story out there and putting yourself out there and how you feel about that. This is coming from that a bit, but a little bit more specific as far as your child's consent for being this representation of their disorder, and how you feel as far as putting these difficulties your child experiences, these milestones, so they can't say it's okay for you to do that. How do you come to some sort of an okay place with yourself as far as not having your child's consent but putting them out there anyways?
It's a good question. I think as far as test goes, it's not the kind of thing that we can really discuss with her. She's 9, but her intellectual disability kind of leaves her more at the level of about a 2-year-old. So she doesn't know any of this stuff that we're doing. She's just not in that place. And our feeling is it is putting her face all over the place, and we struggle with it. I mean, we do. It's something that is, you know, we're getting better about it, and we've been doing it for a long time, but we're not 100% comfortable with it all the time. She has an older brother, and he really doesn't like to be as out there, and so I've thought about that a lot. I don't emblazon everything I do with his picture. I definitely kind of try to keep him out of it. If I talk about him on the podcast, it's just kind of in passing, and I really don't reveal anything personal about him because of that issue. I just feel like he's 13 and he doesn't want to stick out, and I get that, and so I leave him out of it whenever possible. But that's a good question. Thank you.
Thank you. Well, thank you so much, Bo and Rich, for your information, for sharing your story. This has been incredible. Before heading out, we'd appreciate it if you open up your app and take a brief survey on this session. The feedback is invaluable, and I know Global Genes appreciates it. If you still have some questions for our speakers, we're going to be moving into our networking break, sponsored by Leadiant Biosciences, and ask that if you would like to chat with them, we're going to move outside to the break. But a few extra reminders before we head out. We have a guided networking session on telling your story and promoting your community, hosted by Travis Flores and Madison McLaughlin. I suggest you check that out. It's downstairs in the Nautilus room. We are moving on to our networking also open networking in the exhibit hall, sponsored by PRA Health Sciences, where you can visit with our exhibitors and hear from our poster presenters, sponsored by Aldebaran. Sorry. Our next breakout session will begin at 3 p.m. Thank you again for joining us, and thank you so much, Bo and Rich.